We know blood capillaries are basically the exchange vessels. That's the place that will let substances get out of the blood capillary, go to the tissue, or from tissue get into the capillary. There are different strategies that we use to allow substances to be exchanged between blood capillaries and cells that we have outside. However, in this video, we focus on a strategy that is called bulk flow. When we say bulk flow, what we mean is large numbers of ions, molecules, and particles that we have in fluid all together move from high to low pressure. So it is important to note that bulk flow is not about movement of a particular ion or molecule. It's about a fluid that has lots of ions, molecules, and particles inside. That's why throughout the video I refer to bulk flow as moving water and solute from high pressure to low pressure. And when we focus on our blood capillaries, we see that bulk flow could happen in two different directions. Either from blood, water, and solute are pushed out into the interstitial fluid. If that's the case, we name this filtration. Or water and solute from interstitial fluid are pushed back into blood. This type of bulk flow is called reabsorption. So the direction that water and solute are pushed would be different between filtration and reabsorption. We do have pressures involved because we say that bulk flow, filtration, and reabsorption are movement from high to low pressure. Two pressures help filtration. Basically, these two pressures are pushing water and solute out of blood capillaries. One is blood hydrostatic pressure, the other one is interstitial fluid osmotic pressure. Recall that blood hydrostatic pressure is generated by the contraction of left ventricle. When left ventricle pushes blood into the arteries, when blood flows through these blood vessels, it pushes the wall of the blood vessel. So imagine when blood flows into this blood capillary, it keeps pushing the wall. And when I focus on the arrows, I see that blood hydrostatic pressure is pushing water and solute out of the blood capillary. Interstitial fluid osmotic pressure, one easy way to think about it is that in interstitial fluid we do have solutes, and we know that solutes attract water. Basically, they pull water out of the blood capillary. So both of these two pressures are outward. They help promote filtration. The two pressures that help reabsorption, basically they push water and solute back into blood, would be blood colloid osmotic pressure and interstitial fluid hydrostatic pressure. Let's start with this one. In reality, interstitial fluid, which is around the blood capillary, is a fluid, and it applies some pressure, even though it's a very small amount of pressure, on the wall. So it's very easy. Blood from inside is pushing the wall, interstitial fluid from outside is pushing the wall. And that would be interstitial fluid hydrostatic pressure that basically help reabsorption. Blood colloid osmotic pressure is because of the solutes that we have in plasma. Recall that in plasma we have also solutes including proteins, and some of those proteins are very large proteins. These solutes are attracting, pulling water into the blood. Now that we know we have two pressures, that they're outward, they promote filtration, and two pressures are inward, they promote reabsorption. To find out exactly in a blood capillary, are we going to have filtration, we're pushing out, or reabsorption, pushing in, we just need to find the net filtration pressure, which we measure it always by this formula: outward pressures minus inward pressures. And it's interesting that in our blood capillaries, when we go to the beginning of the capillary, which is close to the arteriole, we see that here in this spot, outward pressures are stronger, so filtration takes place. But when we get to the end of the blood capillary, the one that is close to the venule, we see that in this place, inward pressures are stronger. So at the end of the capillary, reabsorption happens. So in general, at the beginning of the capillary, water and solutes are pushed out, and at the end of the blood capillary, a high percentage of what has been pushed out will be pushed back in.